Hello everyone, welcome to Just Biotech Geeks. In this tutorial, we are going to see how to prepare a protein and ligand for molecular docking. I will be providing the link for downloading all the softwares as well as the link for downloading the protein and ligand structure in the description of this video. You may also join our Telegram channel for downloading the softwares. So, let's begin by installing the softwares required for protein and ligand preparation. Here, I have already downloaded some softwares required for protein and ligand preparation. Now, you can see the folder for SPDV Viewer software. We need not install SPDV Viewer software as we can launch it directly. Next, we must install OpenBabel. For installation of OpenBabel, click on the installation file and click Next. Click on Agree, press Next, click Install, click on Next, click on Finish. Again, click on Finish. Here, you have successfully installed OpenBabel in your computer. Now, you can see the software. We will be using OpenBabel software for converting one file format to another file format during ligand preparation. So, for time being, let's close this software. Now, let us open Google Chrome. So next, we will be downloading the 3D protein structure from the Protein Data Bank. Before downloading a protein, we must refer to various research articles and select the appropriate protein based on the field of study and interest. Protein structures can be downloaded from the PDB database. The PDB database is a protein data bank where you can download the 2D and 3D structure as well as various information and sequence related to various proteins. So, let us search for PDB in the search tab. Let's click on the first link available here. Now, here we will be searching for the desired protein of our interest. I would be focusing on human papilloma virus HPV 16 E6 oncoprotein, which is responsible for cervical cancer. Here, every protein is assigned with a 4-letter unique code. Based on the research article that I have referred to, I would be downloading the 3D structure of 2FK4. Here, you can see various information related to this particular protein structure. Now, just click on Download File and click on PDB format. Here, you can see the protein structure is being downloaded. Now, let us go to the particular folder where the file has been downloaded. Now, let's move the file to a separate folder. Here, I have already created a separate folder for protein. Now, open it using WordPad. Here, you can see the coordinates related to the protein. Next, we must delete the heteroatoms from the file. So, here you can see a heteroatom over here. In some case, if there is more than one heteroatoms, you must delete all the heteroatoms. Now, let us select from heteroatom till just before the end and delete the particular data. Now, you must save this file and close it. Next, we must launch SPDB Viewer from the folder. You may close this tab. Click on the file, open the PDB file, go to the particular folder and select the particular protein file and press Open. Click on OK. You may close this tab. Here, you can view the 3D structure of the protein. Next, we are going to do the energy minimization step. For doing energy minimization, let us click on Select and select all. Now, Let us click on the tool and click on energy minimization. This process might take some time. Make sure that you don't access or open any other files or softwares in the background. Here the process is completed. Now you can see the values over here. Now you may go to File, click on Save and click on Current Layer. Now rename it as 2fk4 underscore prepared.pdb and click on Save. You can name this particular file with any name of your interest. Now you may close this file. Next, open the prepared protein with WordPad and scroll down. Here you can see some coordinates that were formed when we used SPDB Viewer. Now you must select the data, make sure that you leave the end alone and press delete. Now save this file and you may close it. Here our protein is ready for docking. Next, let us move to ligand preparation. For ligand preparation, we can download a ligand structure from various chemical databases such as PubChem, DrugBank, etc. So here, I would be downloading the structure of curcumin from the PubChem database. Now, let us open a new tab and search for PubChem. Click on the first link. Let us search for curcumin in the search tab. Here, you can find the structure of curcumin and all the information related to it. You may either download the structure from the database or draw the structure using ChemSketch. I will be demonstrating how to draw a chemical structure using ChemSketch in the other tutorial. As of now, we will be downloading this particular 3D structure in SDF format. Let us click on Save. So here, the structure of curcumin is being downloaded. Now, let's minimize this tab and go to the download folder. Move this file to the ligand folder which I have already created. Now, we must search for OpenBabel in the search box and launch it. Here, select the file format as STF. Now, select the particular ligand file that we have downloaded from PubChem and click on Open. Next, we must select the output format as PDB from this drop-down list. Now, select the folder where you want to save the converted file. So, I will be saving the file in the ligand folder and naming it as 1.pdb. Click on Save and now click on Convert. Here you can see the converted file. Now, we can use this ligand structure for docking. That's all for this video. Watch part 2 on setting grid parameters in molecular docking. Thank you.